Allergy can be treated by inhibiting either immunoglobulin E production or the effector pathways activated by cross-linking of cell surface immunoglobulin E. There are two primary clinical strategies for the prevention and treatment of allergy. One is desensitization, and the other is blockade of the effector pathways. There are also several approaches still in the experimental stage. In desensitization, the aim is to shift the antibody response away from one dominated by immunoglobulin E toward one dominated by immunoglobulin G. The latter can bind to the allergen and thus prevent it from activating immunoglobulin E-mediated effector pathways. Patients are injected with escalating doses of allergen, starting with tiny amounts. This injection schedule gradually diverts the immunoglobulin E-dominated response driven by Th2 cells to one driven by Th1 cells, with a consequent downregulation of immunoglobulin E production. Recent evidence shows that desensitization is also associated with a reduction in the numbers of late-phase inflammatory cells at the site of the allergic reaction. A potential complication of the desensitization approach is the risk of inducing immunoglobulin E-mediated allergic responses. An alternative and still experimental approach to desensitization is vaccination with peptides derived from common allergens. This procedure induces T-cell anergy, which is associated with multiple changes in the T-cell phenotype, including downregulation of cytokine production and reduced expression of the CD3 T-cell receptor complex. Immunoglobulin E-mediated responses are not induced by the peptides because immunoglobulin E, in contrast to T cells, can only recognize the intact antigen. A major difficulty with this approach is that an individual's responses to peptides are restricted by their MHC class II alleles. Therefore, patients with different MHC class II molecules respond to different allergen-derived peptides. As the human population is outbred and expresses a wide variety of MHC class II alleles, the number of peptides required to treat all allergic individuals might be very large. And that's allergy can be treated by inhibiting either immunoglobulin E production or the effector pathways activated by cross-linking of cell surface immunoglobulin E. Have a great day.